Right, so welcome to the second session. So this one is for separate biology students only. So just a reminder that if you don't do separate biology or separate science, then you do not need to watch this video. Okay, so we're going to go through exactly the same as what we've just done for the combined science, but we're going to look at some questions specific to separate biology. Okay, so we're going to start with this question here. So it says, a student is given a tube containing a liquid nutrient medium. The medium contains one type of bacterium. So it says clearly in this question you will be assessed using good English, organising your information clearly, and using specialist terms where appropriate. So again, just like we went through in the last questions, you can bullet point your information in this answer. Okay, so we're going to hug this question and we're going to break it down before we look at what the answer would be. So it says the student is told to grow some of the bacteria on an agar jelly in a petri dish. Describe, which is our key word, so describe how the students should prepare an uncontaminated culture for the bacterium in the petri dish. You should explain the reasons for each one of your steps. So because it's saying describe how the student should prepare, this one is asking you for a method. Okay, so we need to be writing a method of how we would do this. Below it says you should explain the reasons for each step. So every time you mention a step in this question, you need to explain why it's really important that you've done that step. Okay, so if we look at the question, it's asking us about bacterium. Okay, so bacterium is a pathogen. So we already know that. And it's told us two pieces of equipment that we're already using. So we've got our agar jelly in our petri dish. Okay, so we need to look at how we're going to set this question up. So first of all, we're going to start by the pre-inoculation. So what do we need to do before we start this experiment? Okay, so the first thing we would do is we would make sure, and I'm going to show you the mark scheme for this so we don't have to keep writing it out actually. So if we look at this, I've broken this down for you so you can see which bits are the steps and which bits are the explained reasons. So pre-inoculation, petri dish and agar sterilised before use. So this bit is our step. Directly underneath, we've explained exactly why we've done that step. So to kill any unwanted bacteria. Again, the step, inoculating lupus pass through the flame or sterile swab, explaining why to sterilise or kill any unwanted bacteria. Okay, so that's pre-inoculation. Now during the inoculation, it's exactly the same again. So what steps do we need to take and how are we going to explain them? So the first step, the loop or swab is spread onto the bacteria agar. Then what you would do is you would put the lid onto the petri dish and then explain why to prevent the microbes from entering. The final part of it is the post-inoculation. So what do you have to do? So seal with tape is the step, and then explaining why to prevent the microbes entering. Incubate, explaining why again to allow bacteria to grow. Okay, so if you mention each one of the steps and explain every reason why, then that should get you 6 marks. Okay, so if we look at the levelling for this question, here it says, for 5 to 6 marks, clear description of all 3 stages and explained at least 2 of them. Okay? So, we'll move on to a question which is linked to this. So this question here is directly linked to the practical. So, after the culture had been prepared, the student added 1 drop of 5 disinfectants onto the culture. The diagram shows the appearance of the petri dish 3 days later. So again, we're not going to ignore the diagram. So we can look at what our diagram is showing us. So we can see we've got where the disinfectant was in our petri dish, which is here. It's this inside circle on all of them. And then if we look at the diagram, it's told us where there's no bacteria growing. So no bacteria growing is this area around the outside. And then it shows you clearly where the bacteria is growing. So, first question, There are areas on the agar jelly where no bacteria is growing. Why could this be? Okay, so this is basic recall question. So why could this be? Well, that might be because the bacteria has been killed or destroyed. So just 1 mark, so all you have to say is one thing, so killed or destroyed. So, moving further down, the student concluded that disinfectant D would be the best thing for using at home. Give one reason why the student might be correct. So if we look here, disinfectant D is this one here. So he concluded that it might be the best for using at home. Why might this be? Well, if we look at the area around it, where no bacteria was growing, we could say that it has got the largest area or largest space. So the largest area around it with no bacteria. Okay, so next question says, give one reason why the student might not be correct. Okay, so you've really got to think about this one. So why he might not be correct is that this one could be any reason so harmful. You could have that it might work differently with different bacteria. Just shorten that down so I don't have to write it out. Okay, or you could also have disinfectants, maybe different concentrations. So there's plenty of answers you could have. There's a long list here. So if I show you some of the other answers that you could have. So you can look at any of these here. So it could be harmful to people, animals, to the surface. We've said this one. Different concentrations, might not last as long. So 2 marks, but plenty of answers available for you to give, okay? So, moving on. Question 2 that we've got. This one is on a topic which we think, well, came up last year?
Yeah.
Came up last year, so we're thinking it might not come up this year, but it's always good to cover it, so always good to revise it just in case. So this one is on monoclonal antibodies, and it says, these are used to measure the levels of hormones in the blood. Pregnant women produce the hormone HCG, and HCG is secreted in the urine. Then we have figure 1 below. It tells us quite a lot of information in figure 1. So we've got down here, invalid test result, no line appears in this window. Negative test result, the line will appear in this window here. So we can see we've already got 2 lines there in the window. Positive test result, the line will appear in this window here. Okay, so in the control window, but also, don't ignore this bit, in the result window as well. So it says, which test strip shows a negative test? So if we go back up and look at our actual diagram, okay, so which test strip shows a negative test result? So a line that only appears in the control window. So we're looking for just a line in the control window and nothing else. So that's this one here. So all we do is tick the box, okay? Next one. Monoclonal antibodies are used for pregnancy testing. Give one other use of monoclonal antibodies. So this is another basic recall question. So if I show you the mark scheme for it. So it's just this bit here. So a recall of a use. These are the answers you could have had. So identify, locate specific molecules or hormones, locate blood clots, diagnose and treat cancers. So a recall question. Right, so one question which we do want to focus on during this session is this one about plant diseases, okay, and TMV. we think that this one could be, or this could be quite high likely of being tested on TMV on any plant diseases during this year's paper, because it didn't come up last year. So we're going to spend a little bit of time just looking at this question, okay? So TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Explain why plants with TMV have stunted growth. So, explain is our command word in this question. So before we start, we'll go through and we'll underline some key parts of the question that we already know and see if we can work it out. Okay, so TMV can cause plants to produce less chlorophyll. So it's less chlorophyll. So chlorophyll, we already know, is found in the chloroplasts, which are in the leaf. And we know that they are responsible for photosynthesis. So they absorb light for photosynthesis. And during photosynthesis, we know that plants produce glucose, which is the sugar. So, how might the plants then use this glucose which they produce? So they can use it in respiration. And that will produce the energy that it needs to grow. They can use it to produce amino acids. So if you can't see this, I've decided to write all on one side. And amino acids go on to produce proteins. And they can also use it to produce cellulose. Okay, so we've wrote everything down there that we know, just links from chlorophyll and about photosynthesis and how it uses the glucose. Now it says this causes the leaf to discolour. Explain why TMV cause stunted growth. So what we need to do is we need to reverse what we've just looked at. Okay, so less chlorophyll means that less photosynthesis will take place. That means that less glucose, which is the sugar, will be produced or made. That means the plant will do less respiration, so less energy will be produced. And if it's doing less respiration, then we're going to have less energy for growth, less energy to produce our amino acids and proteins, and less energy for cellulose. So you already know all of the key facts. It's just about reversing the answer around. So I'll write this out for you. So less photosynthesis, less glucose produced, less energy for respiration, so therefore less amino acids, proteins, etc. So all these which are down here. Okay? So 4 marks. The first part of that question was this question here. Okay, so it introduced you to tobacco mosaic virus. It told you that it was a disease which affected the leaves. And then underneath that, if we go here, are again questions linked to that. So all tools should be washed in disinfectant after using them on plants infected with TMV. Suggest why. So suggest why means we might not necessarily have taught you this when we did this topic. It's you suggesting a reason why. So the logical reason for washing anything when linking it, when using it with diseases, is either to kill the virus or prevent spread? Scientists produced a single plant that contained a TMV-resistant gene. Suggest how scientists can use this plant to produce many plants with TMV-resistant genes. Okay, so this is a method. So it's a suggest how question. So it might be something that you've learned in a different topic. So you would take a cell from the meristem or use tissue culture. Remember, if something is resistant, it means that it is not affected by TMV. I apologise because my writing is terrible now. So, one more question to go through. So some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in fruits. So glucose is our sugar. So we need to be thinking of a test that we might have done in science to test for the presence of glucose. And it's just a simple description, pick my highlighter back up, of that test. So we're going to start by saying what we would use. So it's Benedict's solution. And then we just state what the positive result for that test would be. So it turns from blue to orange. So 1 mark for saying what test it is and 1 mark for saying the positive result. So, thank you for listening to our Separate Science video live stream. So we hope that's been an extra useful revision for you. And if you need any more revision, we'll leave the comments open or just comment on the video and we'll try and get back to you in the next couple of minutes.